Hi everybody, this is Lori. Before we really get down into the weeds on the topic of hyperlipidemia, I thought it would be helpful for you to have a basic overview of the lipids involved and the different types of lipoproteins that transport them throughout the body. When an individual is being tested to see if they have hyperlipidemia or hypercholesterolemia, as it's sometimes called, a blood test is performed. The blood test looks primarily at two separate measurements in terms of lipids. It looks at the level of total cholesterol and it looks at the level of triglycerides, where total cholesterol is actually the sum of the HDL, LDL, and VLDL cholesterol in the blood. And we'll get more into detail on these terms later in the presentation. Now, when it comes to these two types of lipids, cholesterol and triglycerides, it's really important to realize that cholesterol and triglycerides are two entirely different types of lipids, which we'll see on the next couple of slides. And that's why they're measured and reported on separately. On this slide, we see the molecular structure of a triglyceride, the big picture here, and I've also included a smaller picture of a molecular depiction of cholesterol here in the inset picture, so that you can see that they are in fact two very different types of lipids. The triglyceride consists of a glycerol backbone, this region here, with three fatty acid chains attached to it, these guys coming off here. Now, the fatty acids are chains of carbon and hydrogen, and they can vary in length. Thus, we get the terms short chain, medium chain, and long chain fatty acids that most of us are familiar with. Triglycerides come from dietary sources of fat, and they're stored in adipose tissue until they're needed by cells for energy, at which point their release from the fat cells is triggered by hormones. Thus, they're essentially energy storage molecules. But because they're lipids, triglycerides are insoluble in water, and therefore they do require the assistance of lipoproteins to travel around in the bloodstream. And we'll talk more about lipoproteins in a few minutes. And here you can see the molecular structure of cholesterol in a more detailed depiction. Now, it is a sterol, as you can see here from its steroid ring structure, and its hydroxyl or alcohol group here. And just as a side note, cholesterol is the animal sterol in our diet. We also have plant sterols in our diet, which are called phytosterols. Now, cholesterol also has a hydrocarbon tail, which is this region up here. And although the hydroxyl region of the molecule down here is water soluble, both this ring region here and the tail region are water insoluble. Thus, this makes the molecule insoluble in water as a whole. And so, just like triglycerides, cholesterol also will not dissolve and travel on its own in the bloodstream. It must also have a lipoprotein carrier. Okay, so what is a lipoprotein carrier that we keep hearing about? Well, there are four major types of lipoproteins: chylomicrons, VLDL, LDL, and HDL. And here on this slide, we can see a basic, just generic structure of a lipoprotein, which is basically a complex, or it's often referred to as a particle, of lipids and proteins, where you have a core of lipids here inside, and that core of lipids is encased by a phospholipid membrane out here, and that membrane is interspersed with proteins. You can see these purple guys here. These are the proteins that occur sporadically within the membrane. And since the lipids are water insoluble, this particle here is the, quote, water-friendly vehicle, if you will, by which they travel around in the blood and the lymph. And here you can see they do on this slide, we do have the labeled different types of lipids here. We have our triglycerides, and down here we have, well, we also have phospholipids in the membrane, and then here we have cholesterol, and it's shown in its two different forms. Free cholesterol is basically in its raw form, is how you can think of it when it's picked up. And then once it's picked up by the particle, it can be converted into what's called cholesterol esters. And it's essentially just bound with fatty acids. And what that does is it enables the cholesterol in the form of cholesterol esters to be packed more tightly inside the lipoprotein particle so that the particle can actually carry around more of it. Now these proteins that occur within the lipoprotein's membrane, here these purple ones, are called apolipoproteins. And they are the component that actually enables the lipoprotein to interact with cells at any given destination point in the body, such as a hepatocyte in the liver or an endothelial cell inside a blood vessel, or perhaps even a muscle cell. And the important thing to keep in mind here is that the four different types of lipoproteins, chylomicrons, VLDL, LDL, and HDL, differ from each other in two significant ways. Number one, they differ in their lipid to protein ratio. And number two, they differ in the types of apolipoproteins occurring on their membranes. And the apolipoproteins have names like apolipoprotein A, apolipoprotein B, apolipoprotein C, etc. Or sometimes it's shortened and the terms are ApoA, ApoB, ApoC is another way to say those. Now, when it comes to distinguishing between good and bad cholesterol, the ApoA and the ApoB are the big differentiators, as we're going to see in a few minutes. Okay, so a neat little analogy that I came up with is to think of these lipoproteins as taxicabs. The apolipoproteins on their membranes that we just talked about are depicted here as the red light here on the roof of the cab. And that tells us a lot about the type of cab that it is. And it also gives us an idea as to what its typical passenger load looks like. Now, each type of cab has a specific job to do. It is programmed to pick up a specific mix of passengers and to take them to a specific destination. And you'll see what I'm talking about over the next few slides. Of the four types of lipoprotein particles, we're going to start by looking at the chylomicron. But before we go into detail about the actual particle itself, I thought it would be good to quickly review where they come from. So here we can see up at the top part here, we can see the lumen of the small intestine, where the dietary fats get absorbed and converted to triglycerides by the enterocytes. And here we see our enterocytes of the small intestine. This should be familiar to all of us from Physiology 1. So we have the lumen of the small intestine and the enterocytes, and then the triglycerides that come in from the diet. Because they're not water soluble, they get packaged into chylomicrons before they're secreted. Down here you can see that happening, so that they can be picked up down here by the lymphatic system and then subsequently transported to the circulatory system and then eventually to the liver for processing. So this slide should look somewhat familiar to all of us, as we've seen this before. So here's our very first type of taxicab, the chylomicron. And you can see the basic lipoprotein structure with the core of lipids in the center here that's enveloped by this phospholipid membrane on the outside. And we also see that the major protein component of the chylomicron is this guy here, the ApoB protein. So the light on top of this particular taxicab, so to speak, is ApoB. Now, although it's not depicted in this drawing, chylomicrons are known to possess ApoA proteins as well, but those ApoA proteins are quickly transferred to HDL particles once they reach the bloodstream. In terms of composition, a chylomicron is about 85 to 88% dietary triglycerides, about 8% phospholipids, about 3% cholesterol esters, and about 1% dietary cholesterol, and only about 1 to 2% protein, which is not a lot of protein. Because they have such a high lipid to protein ratio, they are very low in density and very large in size. In fact, the chylomicron is the largest of the 4 types of lipoproteins. So the chylomicron enters the scene from the small intestine carrying these dietary lipids, which is why this particular type of taxicab is so loaded down with triglycerides as its passengers, because it's coming from the diet from the small intestine. And the chylomicron's main job is to transport these dietary triglycerides, which are energy rich, through the body and deliver them to the cells that want to use them for energy, like working muscle cells as an example. Any triglycerides that remain in the taxicab at the end of the so-called muscle route get dropped off at the adipose hotel, where they're stored in adipocytes or fat cells. Our next lipoprotein taxicab is called very low density lipoprotein, or VLDL. Here again, the primary protein is the ApoB protein. We see it depicted down here as B100. They also have numbers assigned to them at times. So the light on top of this cab is ApoB. And here again, we have a lipoprotein that is triglyceride rich. In terms of composition, a VLDL particle is 50 to 55% triglycerides, 18 to 20 phospholipids, 12 to 15 cholesterol esters, 8 to 10% cholesterol, and about 5 to 12% protein. So a little more protein in this one than the chylomicron. But this is also a rather high lipid to protein ratio, thus the name very low density lipoprotein, although not as low density as the chylomicron. So the VLDL taxicab enters the scene from the liver because that's where it is synthesized. And once it enters the bloodstream, its job is to deliver the energy-rich type triglyceride to cells, just like the chylomicrons. And upon reaching a hungry cell, both the chylomicron and the VLDL are stripped of their triglycerides by means of an enzyme called lipoprotein lipase, which is found on the cell's surface. This enzyme breaks down the triglycerides so that they can then be taken up and used by the cell or stored by the cell if they are in fact dropped off at the adipose hotel, so to speak. Now, as VLDL particles are stripped of their triglycerides, bringing down that lipid to protein ratio, they begin to transform into a particle that is more dense. These slightly more dense particles return to the liver, where they are reprocessed and transformed into low density lipoprotein, or LDL, which brings us to our next slide, which is our third type of taxicab, and that is low density lipoprotein, or LDL. Again, you can see the basic lipoprotein structure here, and again, the primary light on this taxicab is the ApoB protein, seen here, ApoB 100 again. And it's important to note that whenever a person's blood is tested for cholesterol, the presence of ApoB protein is most associated with this particle, the LDL particle. In terms of composition, an LDL particle is 10 to 15% triglycerides, 20 to 28% phospholipids, 37 to 48% cholesterol esters, 8 to 10% cholesterol, and 20 to 22% protein. So essentially, an LDL particle is actually a VLDL particle that has offloaded a bunch of its lipid passengers. Thus, it goes from being very low density to just low density, if that makes sense. So, and you can see what happens with the numbers here and how they're starting to shift. The ratio is starting to shift. Now, the primary job of the LDL taxicab is to transport cholesterol, both dietary and endogenous, to cells throughout the body so that it can either be used as a structural component of their cell membranes or be converted to steroid hormones. So we finally come to the fourth lipoprotein, the one that everybody calls the good guy. This is high density lipoprotein, or HDL, as it's commonly referred to. Here, once again, you can see the basic lipoprotein structure as well as the various apolipoproteins that are peppering its membrane. However, unlike the previous three lipoproteins that all had ApoB as their primary proteins associated with them, HDL, conversely, has ApoA protein as its major protein component. You can see three of those here. ApoA here, and we've got an ApoA here, and there's four of them shown here. An ApoA here, no, three. These three yellow ones. So going back to our little analogy, the HDL taxicab is easily identified because it has this ApoA light on its roof, so to speak. HDL is primarily synthesized in the liver, but in recent studies, it has been shown that some HDL particles may actually be assembled by the enterocytes in the small intestine, which is an intriguing concept when you consider that this could potentially lead to some new alternative therapies for upregulating this, quote, good cholesterol in the body. Now, HDL's specific job, once it is synthesized and secreted into the bloodstream, is to go out and pick up excess cholesterol from the cells and transport that cholesterol back to the liver, where it's converted into bile salts and excreted from the body in the bile. This process is known as reverse cholesterol transport. One thing I'd like to point out here is that when the HDL particle is initially secreted by the liver, it's considered a precursor particle or an immature HDL particle. And then as it travels around in the bloodstream, picking up its passengers, i.e. the excess cholesterol from the cells, it becomes what we call a mature HDL particle. In terms of composition, the HDL particle is only 3 to 15% triglycerides. It's 26 to 46% phospholipids, 15 to 30% cholesterol esters, 2 to 10% cholesterol, and about 55% protein. So you can see the protein here is higher, the ratio is higher. The amount of protein to lipid is higher than the other particles. I won't spend a lot of time on this slide because HDL isn't really a factor in hyperlipidemia because it is the good cholesterol, so you want to have lots of it. But I thought it would be good to just mention some of the protective actions of HDL because it is an important part of maintaining wellness when you're talking about lipid levels in the blood. The proteins found in an HDL particle participate in activities like lipid metabolism, free radical scavenging, complement regulation, and proteinase inhibition. Plus, they help modulate inflammation and they scavenge excess cholesterol, as was just mentioned. So these are some of the protective benefits of this particle. Now that we've explored the different types of lipoproteins, let's take a quick look at the difference in their sizes. Notice how large the chylomicron is compared to the other lipoproteins. It's huge compared to those other ones. In reality, though, a chylomicron is only about 1/10 the size of a red blood cell. So just to give you some perspective here. The basic rule of thumb here is that the more lipid the particle contains relative to protein, the less dense it is. And the less dense that it is, the bigger it is. Okay, so now that we have a better grasp on the types of lipids and their lipoprotein carriers, we can better understand a person's lab results whenever they have a lipid profile performed. Let's take a look at what's actually being reported. Whenever we see total cholesterol, we know that they're measuring all of the cholesterol in all of the lipoprotein particles, regardless of type. Likewise, for triglycerides, which is often abbreviated as TG on a lab report, it's a measurement of all the triglycerides in all of the lipoprotein particles, regardless of type. Although most triglycerides are found in the VLDL particles. For HDL cholesterol here, we are, which I want to mention here too, you'll see HDL cholesterol often depicted as HDL-C, which just means HDL cholesterol. It's a little different. This time we're measuring the amount of cholesterol in HDL particles only. And for LDL cholesterol, which is depicted LDL-C oftentimes, we're looking at the amount of cholesterol in LDL particles. However, this number, LDL-C, is actually derived from a mathematical calculation most times, where they're using the first three numbers here on the slide to calculate this fourth number, the LDL-C. So it's typically not an actual measured number value. It's usually a calculation. This slide shows a few extra values that may be reported as part of a lipid profile test. These aren't always included, but sometimes they do what's called an extended lipid profile, and so you might see these on the lab report as well. Here we see the VLDL cholesterol, denoted as VLDL-C, which is calculated based on the triglyceride number on the previous slide, combined with the known typical composition of VLDL particles. And sometimes you'll also see non-HDL cholesterol, this second one here on the lab report, which is also a calculated number. And it's calculated by taking the total cholesterol minus the HDL cholesterol, which makes sense if you think about it. The third one is cholesterol to HDL ratio. And it's simply the ratio, just what it says. It's simply the ratio of total cholesterol to HDL cholesterol. And one other value that may be reported is this fourth one, this low density lipoprotein particle number, which is denoted as LDL-P. And this number represents an actual measurement of the number of LDL particles rather than measuring the amount of cholesterol in the LDL particles as we saw on the previous slide. It's now thought that an interesting little fact that I found is that it's now thought that LDL-P may actually be a more accurate predictor of heart disease risk than LDL-C or LDL cholesterol in certain people. I thought that was interesting. Okay, so don't laugh, but this slide is my own little clip art creation that shows a very simplified depiction of what is going on with all of these lipoproteins. Although now that I look at it, it really doesn't look too simplified, does it? I guess it just speaks to how complex these mechanisms are. But anyway, you can see each of the four lipoproteins that we just covered. And you can see where each one originates and what each one's overall function is in terms of transporting cholesterol and triglycerides throughout the body. So here we've got the liver and we've got our HDL. It's synthesized in the liver, coming out here, going out into the body to pick up that excess cholesterol and bring it back. And that's shown here. Here's our body cells. the HDL picking up the excess cholesterol, taking it back to the liver. We've got our chylomicrons here, which are formed by the enterocytes in the small intestine here when we eat the big fat hamburger. We've got our triglycerides, our dietary fats, and they are packaged in the chylomicrons. This IDL here, I'll just mention, that's just an intermediate form between the VLDL and between the chylomicrons and the formation of LDL that we mentioned on the one slide. But the other thing that's shown here is how the VLDL and the chylomicrons both carry the dietary fat, the triglycerides, to muscle tissue here and down here. And what's not used by muscle tissue is then taken to what I called the adipose hotel and stored in the adipocytes. So I've just captured the real basics here on this slide of what's going on with the particles and where they're coming from and where they're carrying the fats, the lipids. And this last slide, which I did not create, shows an even simpler schematic that compares only the LDL and the HDL lipoproteins in terms of what their jobs are. So here we've got the LDL in the red and the HDL, the good cholesterol, in the green. And this is very simplified, just showing the route here that, the dietary fats and that the LDL cholesterol carries the endogenous and the dietary to the body cells. And then the HDL is bringing it back from the body cells into the liver, broken down into bile salts and into the bile, and then excreted from the body. So that's just another way of looking at these two particular lipoprotein particles. So thanks for watching, and I hope this was helpful.